Thank you very much for tuning in to today's Pineland Speaker Series presentation. We're very fortunate to have with us today Dan Duran. He's with Rowan University. He's a scientist there, and he is going to be talking to us today about tiger beetles and how they are a model for biodiversity in science, which is a great topic. Some people might say tiger beetles, pinelands, what's the connection? One of the first things you learn about the pinelands is you do a lot of reading of Howard Boyd. And Howard Boyd was a researcher who spent a lot of his life learning about tiger beetles. And one of the reasons he came to the pinelands was because of the population of the tiger beetles. So with that, I'm going to turn it over to Dan, and we're going to learn about, just like I said, tiger beetles as models for biodiversity.
Thanks, Joel. I appreciate that. And yes, actually, speaking of Howard Boyd, he was a really big influence on me. He was an inspiration, and I'm very lucky that I got to meet him. And in fact, apparently the last scientific talk that he came to was a talk I gave about Pineland's tiger beetles. So that was very, very meaningful for me. So yes, today we'll be talking to you about tiger beetles. We'll be talking about tiger beetle identification, biology, and then how they can be used as models for biodiversity science. And so we'll start out with that. We'll get into the basics of what a tiger beetle is and what kinds of cool adaptations they have to survive. And many of them live in extreme environments, and their survivorship is really impressive. How they can live in deserts that can get up to, the ground can be 140 degrees Fahrenheit. We'll talk about using tiger beetles as a model for biodiversity exploration, for finding new biodiversity, and applied ecology, ways to make ecology work for our everyday lives, actually. And then we'll talk about some rare species and conservation. So, first question we might have is, what is a tiger beetle and how can you distinguish a tiger beetle from other beetles? Here's a figure from a recent paper that I wrote showing the incredible diversity, morphological diversity, physical diversity of tiger beetles. And many of them don't look very similar at all, but we were able to figure out how they are all related and how some of this diversity has changed over time and evolutionary time. But you look at this and you say, it's hard to believe that these are all the same kind of group of beetles, which is now a family of beetles. And so one thing that you can always see is the mandibles have multiple visible teeth that are on every mandible. So you can see there's at least 3 teeth on each. Some of them have up to 7 teeth. And then they have a molar region, which is harder to see because it's mostly underneath the upper lip. But seeing those complex teeth on the mandibles is very diagnostic. Other predaceous beetles that are very similar will not have that. And we can see here's a different kind of tiger beetle, and you can see that there are, in fact, at least 3 teeth visible. Here's another one, very different group of tiger beetles. This is a very primitive lineage that split off quite early from all the other tiger beetles. Now this is a group of beetles. This is a type of carabid or ground beetle that is very closely related to tiger beetles, and very often they get confused by even sometimes entomologists. But if you look at the mandibles, you can see that they have sharp cutting surfaces, and they are predators, just like a tiger beetle, but they don't have those multiple simple teeth. These are other beetles that are very frequently misidentified as tiger beetles because their overall body shape and their large eyes, in the case of the one on the right, makes them look very much like a tiger beetle. But again, they don't have multiple visible teeth on those mandibles. So they are not tiger beetles. Now, let's talk a little bit more about what a tiger beetle is, not just how to recognize them. Well, they are fast-running predators of pretty much anything smaller than themselves. They will eat ants, they'll eat little flies, they'll eat other beetles. Sometimes large tiger beetles will even eat small tiger beetles. They're just vicious predators. And they're found in primarily open habitats. Plants get in the way when you run fast. So they're usually going to be found in sandy areas, bare clay, bare rock, things that we could think of as early successional habitats, things after a fire, after wind, after water scouring, places that are going to be kept open. Here's a typical tiger beetle habitat. This is a fantastic place to find a number of species, and many species also like ecotones, areas where you have a transition from one habitat into another habitat. So where it gets grassier or scrubbier, you'll often find tiger beetles in the open sand that's right next to those. So they can run and hide, and they can come out into the open to hunt ants or whatever they want to. And if they need to hide from predators, they can go into the vegetation. This is an area, I was driving by a spot in Mississippi, and I saw this and I said, this is fantastic. This is eroding red sandstone, and just great, great habitat for tiger beetles. Unfortunately, a lot of times people see these open habitats and they think of them as waste places. I see this and I say, awesome. A lot of people see this and think, wow, that could be a housing development, or why don't we turn that into a forest? Well, tiger beetles don't really like forests for the most part, or they like open areas in the edge of forests or trails in forests. But there are a lot of species that live in these open early successional habitats, and we often forget about that kind of diversity. So this is not a waste area. This is great habitat for many species, not just tiger beetles, but tiger beetles are a great example. And like I said, they can live in forest. Here's an example of a trail through a forest in India. And the ones that live in the tropics often do different things. Sometimes some of them will actually live on plants, but they'll live on top of the leaves. So they're still kind of out in the open. They're not hiding in the base of the plants, but they'll actually hunt for small insects on the leaf surfaces. There's a lot more surface area up on those leaves. There are about 3,000 species of tiger beetles globally. New ones are being found all the time. In fact, so far in the last 5 years, I have described 10 new species and a new genus. So there are new species out there still to be found. They're found on all continents except Antarctica and all but the most remote oceanic islands. You don't find them in Hawaii, but you do find them in places like Socotra, places that are, relatively close to a mainland. They have been used as models for studying all kinds of different things, including speciation, understanding how ecological niches partition species and allow them to coexist. Sometimes you'll find a number of species living in the exact same area, and you say, how is that possible? Aren't they all hunting for the same things? Isn't there competition? But there can be differences in mandible shape and size, and they can all be specializing on different prey, or at least that's an idea. It turns out that a lot of the research says they don't care, and they'll eat pretty much whatever they want. And each species is there because they can survive on their own, and competition may not be as much of a factor as previously believed. We've studied their thermoregulation. It's actually amazing how they can survive extreme temperatures that they do. There are species that live 12,500 feet up in the mountains, right next to snow, and other species that live in the hottest deserts in the world. And you say, how can they do this? It's pretty amazing. We'll talk a little bit about how they can do that in a minute. We've used them to understand developmental plasticity. There's all kinds of variations sometimes within the same species when the environmental conditions are different. And so it can change colors, it can change patterns. And certainly we have studied them for their conservation biology potential as bioindicators. And when I say this, I mean tiger beetles apparently are good predictors of other diversity. So if you can actually study the tiger beetles in an area, you have a great idea of how many butterflies there are, of how many birds there are, of how many lizards there are. And so they may be a useful heuristic for understanding an overall diversity of an area, because they correlate very well with other types of biodiversity. Now, they are very popular. They are popular with amateurs, with professionals, with naturalists of all stripes. They're well represented in museums and private collections because many of them are really pretty and many of them are fast, and that makes it exciting to try to catch them. There are a number of field guides written about tiger beetles, and I am shamelessly pitching my field guide here. So I am a co-author of a field guide to the tiger beetles of the United States and Canada, and this is probably the only book that you can have to identify all tiger beetles in the United States. So if you're interested, that's out there. Please get the second edition, not the first edition. But tiger beetles are really, really pretty, and in many cases. And so you can see why these would be popular. I mean, they're basically rainbows, just unbelievably beautiful species throughout the world. We can see on the left an arboreal species of tiger beetle, which is fairly uncommon, that lives in Thailand and other places in Southeast Asia. Then we see the one in the middle is a very large tiger beetle that lives in China, Japan, and Korea, on the ground and trails through a forest. And then we have an African species on the right that lives actually also above the ground. So I've shown a couple of species that are not typical, but they're very pretty. But many tiger beetles are actually quite cryptic. About half of all species look like dirt, like mud, like sand. They can look like the rocks they live on, and they can be, species can live in any of those types of environments, and they can be super, super hard to see in a natural context. And in fact, some of them are so hard to see that you may not even notice them until they move because they blend in so well. Now you look at the one on the right and you say, that looks like that would be easy to see. It's got this bold pattern on it. But in context, it's actually really hard to see. They can blend in with the types of sand grains that they live on, where you can have different types of materials, parent materials like quartz and feldspar and obsidian and all these different things that can be in there, and they can really blend in with those. Now, here is one of our local tiger beetles that I will talk about more later in the talk. This here is the ghost tiger beetle, which is virtually invisible on the ground. In fact, the only thing you can really see sometimes are the eyes, but the eyes can look like flecks of darker materials, so actually they blend in as well. It's really, really difficult to see. In fact, a spectral analysis says these are 98% identical to the wavelengths that are reflected from the ground where they live. So they're really, really hard to see. Yet other tiger beetles are actually going to be mimicking ants, which is really cool. And that's a great strategy in the tropics, because if you look like ants, there are quite a lot of really noxious ants that can bite or sting very hard in the tropics. And so it's a great idea to try to, or I shouldn't say idea, but a great strategy to mimic those types of things. And you can see here's one of the ant mimics that lives in Southeast Asia all the way through to Australia. And another thing that you could mimic is a velvet ant, which is not actually a type of ant, but a wasp. It's a wingless wasp that's one of the most potent stinging insects in the world. And here's an example of a species on the bottom that is a tiger beetle mimicking a type of velvet ant on the top, which you can see there. And having those two big orange spots surrounded by black apparently is a pretty good mimic for that species. Now, talking about their biology a little bit more, they are poikilotherms is the scientific term that we use for cold-blooded. Scientists don't like to say cold-blooded because very often things that we call cold-blooded actually have warmer blood than warm-blooded species. The whole idea of a poikilotherm is it doesn't produce its own metabolic heat. Instead, it gets its heat from the environment around it. So think of like an alligator or a crocodile where they have to warm up by sitting on a beach or rocks where the sun can hit them for a while and it really starts to warm up and they can transfer that heat to their body. Well, again, that transferred heat to their body could actually make their body quite warm. So we don't like to say cold-blooded. But they can, just like a crocodile, they can alter their internal body temperature through their behavior. Get into the sun, get out of the sun, go to a place that's sitting on a south-facing slope. That's a great idea. In the northern hemisphere, a south-facing slope is going to typically be warmer than a north-facing slope because it gets hit by the sun for a longer period of the day. So when these tiger beetles are all warmed up and ready to go and ready to hunt, they're in a foraging posture. Foraging means just simply that they're ready to hunt. They're going to look like this. They're going to have their body slightly elevated off the ground. Their knees are bent. They're ready to run. And some of them can run incredibly fast, over 100 times their body length in just 1 second, which is just incredible. And now when they have to warm up, let's say that they're not warm, they've cooled off, or they just came out for the first time for the day, they will do this basking behavior where they make their body very flat against the surface. Again, that could be a rock or it could be clay, but their legs are going to be splayed out and their shadow is not going to be visible because it's going to be directly under them, since they will be having the sun hit them at the strongest angle possible. So you can see all of these ones are trying to warm up to get ready to be able to do their things for the day, to be able to forage and possibly mate. Now, when they get too hot, and this certainly happens, especially species that live in deserts, that live on white sand, that live on beaches, they're going to need to cool down quite quickly, and they can stilt, is what this is called, where they actually extend their front legs as far as possible, and they get their head off the ground as much as they can, and they can get their head over an entire body length off the ground. And that's really important because their head contains their brain, and their brain cannot get too hot or they could die. So they have to get their body cooler by doing this. And this actually does several things too. Not only does it get their head further off the ground, but it allows air to circulate underneath their body. They can get some convective cooling. They will face the sun, and when they face the sun, they have a big white visor on their face, which actually reflects a lot of the sun's energy back into space and prevents their head from being as hot as it would be. Think of one of those things that you could put on your car windshield that's either white or silver that reflects a lot of the heat back. And they will, because they're facing the sun, they're going to minimize the amount of their body that is being hit by the sun's rays, right? So they're going to keep their body at that angle, which is why the shadow is the smallest it can be when they're in this mode. So all of these behaviors are going to allow them to cool down. Now, if it's still too hot and they can't get themselves cooled down, they better get into the shade. So in the case of some desert areas, there may not be a lot of shade. You have to find the only thing that's out there. I've often found in deserts, if somebody threw an old refrigerator or a tire in the middle of a big desert playa, you will see, if it's a really hot day, you go out there, in the shadow, there will be tiger beetles all in big congregations in the shadows. It's pretty neat to see. And if that doesn't work and you're truly in danger of overheating and dying, dig, get underground as fast as you can. It might be the only way to keep your body temperature from being lethal. Now, tiger beetles are food for other things. They're very good at avoiding being eaten, but still some things will eat them. And here we can see, I believe that's a piping plover chick that is eating Ellipsoptera marginata, which is one of our tiger beetles that we have in New Jersey along the beach. They are eaten by birds, not very commonly, but some birds do eat them. They're eaten by large predaceous flies, like we can see here on the upper left. That is a robber fly. Robber flies are the main predators of tiger beetles. They are very well adapted to eat tiger beetles when they fly from the ground. And tiger beetles will fly to escape. And so as soon as they get up off the ground, a robber fly can swoop in and grab it in midair and then fly back to a perch, like you can see here, where they will stab it with their mouth parts and then eat it. So that's something that we can see. Sometimes lizards will eat tiger beetles. We can see one there on the bottom eating, that's a European species, Cicindelidae hybrida. And ground spiders, like wolf spiders and other things, will sometimes hunt tiger beetles and run very quickly out into the open, grab one, and eat it. That is eating a particular species that is very common in New Jersey. That is Cicindelidae sexcutata, the six-spotted tiger beetle. Now, another really cool thing about tiger beetles is they do have some really cool defenses. They are one of the few species on Earth that produces its own hydrogen cyanide from its body. We can see a bunch of things here that all do that, and this is about it. There's some ants that do it. There's some moths that do it, like this type of tiger moth that we see here. There's not centipedes, millipedes that do this as well, and then some tiger beetles are able to produce hydrogen cyanide. So that's a great way to keep things from eating you, is to spray cyanide in their face, right? And we can see here's multiple different kinds of anti-predator defenses that this particular species, which is also in the New Jersey Pine Barrens, only in a very small area where we get more red clayey ground with big pebbles, big coarse pebbles. I've seen them in a few places. I've seen them at Warren Grove. They're there. Anyway, where you get sort of more coarse pebbles and you get some reddish sandy clay. These guys will often live on pebbles, and they can look kind of like the ground. They're brown colored, they're hard to see, but underneath of them is a bright reddish orange abdomen. And that is a warning that they have toxic chemicals in them. When you make them scared, they will fly and they will expose that red. And to most predators, that is a clear signal that this is not to be messed with. And if you do mess with them, they will spray hydrogen cyanide on you. It's not enough to hurt a person. It's extremely small amount, but it is enough to repel a lizard or a kestrel or one of the other predators that they might have. Okay, so let's talk about using tiger beetles as a model for biodiversity exploration and applied ecology. Now, I'm very interested in the evolutionary ecology of tiger beetles and how these different aspects all sort of interrelate. We can talk about their body structures, their color, their shape, So we could say the morphology of them, and think about how that is influenced by the environment, the temperatures, the soil types that are out there, the geographic areas that they live in, and again, how this all connects to their genetics. And there's some really interesting questions we can ask here. And I'm going to show you some ways that I've done research to understand a little bit more about the biodiversity of tiger beetles. So first of all, when you see all these different colors, sometimes you're going to be surprised to learn that very, very different looking forms can be the same species. Here are two different populations or subspecies of Cicindelidae punctulata. We see one from Arizona here in Cochise County in a famous place for tiger beetle collectors called Wilcox Playa, where there's dozens of species out there. It's a really amazing area. And they are bright blue. They are bright, shiny blue. Yet in New Jersey, they're a dark coppery brown. And so that's the same species. And you say, is there a different gene for a different pigment that they have? It turns out that it's not. It turns out that a lot of this variation in color has nothing to do at all with different pigments. And actually, all their colors, every one of these colors that you can see, both the colors here and these colors here, are all caused by one single pigment, melanin. Same pigment we have too. And so what happens is they have these alternating layers of clear cuticle, of clear skin that alternate with dark melanin. And the distance between those layers is going to affect what kinds of wavelengths of light are reflected. And so if they're, for example, 540 nanometers apart, they are green, because that's what green is 540 nanometers. And so you're going to get all these different colors simply by changing the distance between those layers of melanin. Pretty cool. It also means that the environment might change their colors. So going back to this, it may be that these ones are blue because they live in a much drier and hotter environment than New Jersey. And in fact, we've done this. We've taken ones from Arizona and made them grow up in New Jersey, and they look like ones from New Jersey, showing that this is totally not related to genetics at all. And then sometimes it may be related to genetics, but at least in some cases, it's not, in this example that we've shown here. And in fact, there's some extreme variation within a single named species. This is from a recent paper that I just published in January, where we looked at this incredibly variable species. This is all one species, or at least so it was believed. It turns out there are actually two species here. The one in the upper left is one species, and all of these other color forms are just one separate species from that black one. So that's incredible. How can there be so much variation? Well, not only is this variation extreme, but it's not exactly, it didn't partition the way that we thought. So what we learned is all those colors don't really mean very much at all. What matters is where you geographically live. We can see over there in that map, it shows the shaded in gray areas are places where we have populations of the Cicindelidae polychula complex, also known as the limestone tiger beetle, because they live on limestone. And we see that when we look at it, some of these different color forms, and some of the ones that are actually quite dark too, and some of the ones that are quite brightly colored, all live in mountain ranges in West Texas through Mexico and parts of southeastern New Mexico as well. And they are quite genetically different from the ones that live in central Texas up through Oklahoma. So that's interesting. And it turns out that the differences are predicted by when it rains. If you want to know what species you have, it all depends on when it rains. The colors tell you almost nothing, but the timing of their life cycle totally does. So those Western mountain forms are going to be active from July to early August, and the ones in, let's say, the hill country of Texas and parts of Oklahoma, those are going to be active in the fall, in September all the way through to December, early winter. So they're not even out at the same time of year. And that's interesting how that predicts their relationships. And it turns out they're not even each other's closest relatives. That one species I showed you in New Jersey, the one that lives on pebbles in red sands and clays, that's called rufiventris. And the ones from central Texas are more closely related to them than they are to what was believed to be the very same species in those Western mountain ranges. So what we're learning here is your biology may be a better predictor of relationships than the way that you look. And here is a detailed phylogeny using mitochondrial DNA of the entire larger group here, of this whole genus, of all of these different ones here that all have red abdomens and all typically live on rocks or pebbles. Almost all of these ones live on rocks or pebbles. And so we see here that there's some weirdness. There's some strange things. It turns out, like, why are there two things that say arcumatillus in the Polychula clade? Why would that be? Why are there ones? And if you look at the brown area, the Shalpi and Sympatra congeners, which just means other species that live in the same areas, they're all mixed up. There's something weird going on here. And so this is the mitochondrial DNA that's showing this pattern. But when we look at the nuclear genes, when we look at all of the rest of the genetic data, they tend to come out pretty much exactly the way that we thought they would, you know, matching their names, matching the forms that we had described. And then it also does that too down here. This is a little more complex. And we see sometimes there are multiple color forms that all have the same name. But again, we're seeing the names are matching. The names are matching. So what's going on here, especially in this part of the mitochondrial tree where everything is all kinds of mixed up? And yet their nuclear DNA says they go right where they should. So why is the mitochondrial DNA telling us a different story? Well, it turns out that there's a reason why some individuals appear to have the wrong mitochondrial DNA, and it has to do with hybridization and a process that we call introgression that I'm going to describe to you in a minute. I'm going to walk you through how this happens. So sometimes you will find habitats that are exactly intermediate between the requirements for two different species. We see all these limestone rocks here, which is what Polychula likes. That's perfect habitat for them. And then we see more hard-packed sandy clay areas that's more of the habitat that you'll find Shalpi in. And so these trails that humans have cut through these areas of the hill country, you know, may actually be great habitats for both of them to meet. And they are very closely related, and apparently they're genetically capable of forming hybrids. And so along these areas, they may hybridize right in these interfaces. And so I've actually found hybrids in the wild, individuals that are almost exactly intermediate between the other two species. And genetically, we did find out, yes, they are verified to be hybrids. And so what's going to happen here is this. Let's think of it as the nuclear genome, so all of the genes as a pie, right? Let's take a male Polychula, and we'll say it's a blue colored nuclear genome for Polychula, and we'll take a female Shalpi, and we'll say it's an orange colored genome for that. And then let's also look at the mitochondrial DNA. The mitochondrial DNA only comes from the mother. So in the case of the Polychula, we'll call that a P. In the case of the Shalpi, we'll call it an S. Now, imagine that they mate. They come into this area, both species are present, and typically they only mate with their own kind. But in this case, somebody just made a mistake and they mate with each other and they form a hybrid. Now, the genome of that is going to be exactly half of the Polychula and exactly half of the Shalpi. But the mitochondria only comes from the mother. So right now, it's got 100% Shalpi mitochondria. Okay, now this hybrid, which is viable, it's alive, and it is fertile, it can actually interbreed with either of those species, is going to interbreed with one of them. And let's say it interbreeds with a Polychula, right? When they mate, they're going to form what we call a back cross, which means it's 75% Polychula and 25% of the hybrid of the other species, Shalpi. Okay, so now if we look at the nuclear genome of that individual, it's going to be 3/4 Polychula, 1/4 Shalpi, but it still has that Shalpi mitochondrial DNA in it because it can't get rid of it. That's going to continue down that line. So you see where this is going. Just 3 generations of back crossing back into that Polychula gene pool, looking more and more like a Polychula every single generation. It's going to be 94% Polychula. You won't be able to tell that it has any Shalpi in it, but the mitochondrial DNA will remain Shalpi. So this is what's happening. We're finding individuals that are 94, 98%, some very high percentage of one species. So they look just like it, but because of past hybridization, they have the other mitochondrial DNA in them. So that's going to explain a lot of what's going on there. So again, it was interesting to learn more about their evolutionary history and understand why mitochondrial mitochondrial genes may not tell us what we think they're going to tell us. Now, I want to give you an example of using ecology to solve real world problems here. Okay, so when I'm doing this research, sometimes people say, I don't get it. I don't know why you do this research. I mean, how does this benefit anybody? And I understand that that's the way a lot of people think. I think that science is very valuable for us to have a body of knowledge, and sometimes that knowledge that we had no idea why it was going to be useful, it shows up as being really important, and nobody knew that ahead of time, right? So as I'm doing my basic research, my research that doesn't have obvious applications right now, sometimes I find applications just by dumb luck. And this is, again, why we do science, one of the reasons why we do science. So when I was studying this genus of tiger beetles called Dromaeosaurus, I was looking at museum specimens, and museums have such incredible value. There's so much information there that people don't even understand how much there is. And so I'm going through these different specimens, and I was finding ant heads attached to the mandibles sometimes of these Dromaeosaurus tiger beetles. And it was happening quite a lot. And I was sort of surprised because it's not something you see very much. And so if you're wondering why this is happening, well, tiger beetles are vicious predators. They eat ants on a regular basis, but ants are vicious too. And so in the process of hunting an ant, attacking it, and trying to eat it, sometimes they eat the body, and in the death throes, the ant clamped its mandibles onto part of the leg or an antenna or something like that, and it didn't get pulled off. And so the rest of the body got eaten, and now the tiger beetle has a permanent earring of an ant head. So showing this history of this battle that they had in the past. Pretty amazing. And so I knew a great ant expert who was at Drexel University, and now she's at Arizona State University. And I said to her, can you identify what kinds of ants these tiger beetles have been eating just by looking at their heads? And she said, yeah, absolutely. We could totally do that. And so she started looking at quite in detail of the mandibles and parts of the other parts of the face of these ant heads. And she said, you know, one of your ants is Solenopsis invicta. And immediately I remembered that Solenopsis invicta is the red imported fire ant. This is a $6 billion a year pest. And I just learned that my tiger beetles eat them. And in fact, almost nothing will eat them. So this is really, really interesting. This is some newer research that we've done, and I'm looking forward to turning this into maybe a bigger project. But what I wanted to do was I wanted to find out how often do they eat these red imported fire ants? And that's actually hard to do because tiger beetles are like spiders. They don't eat the hard parts of their prey. They actually only suck up the liquid. And so they'll chew them really well, and they'll suck up all the juices, and then they spit out the hard parts. So you can't look in their stomachs and try to find a fire ant in them because you won't do it. But the DNA of the fire ants may be in there. So what I did was I found a recent paper looking at the evolution of ants, and I found how these different types of ants were related. And I looked to see what kinds of ants are found in the same areas where my Dromaeosaurus tiger beetles are. There were chromatogasters, Solenopsis, Monomorium, different types of ants. And the one I really cared about was Solenopsis. And so I wanted to see, can I make a particular genetic primer? We call them primers. These are things that will bind only to the DNA of what you want them to. Could I make primers that would work to be able to identify whether they've eaten a Solenopsis ant, a red imported fire ant? That's what I wanted to do. So what you do is you look for stretches of DNA in these different species of ants that are unique. And then you can make something that will match them. And that way you can find out whether or not you have that type of DNA present inside of your tiger beetle, right? So I looked at all these different things, and I really care about the one that says S invicta. And I was looking for something that was distinctive. And you can see at the position 110, it has a G where everybody else has an A. So that was one thing that was distinctive about it. And in fact, you could find that it would be easy to produce a primer that would match specifically this insect, only the red imported fire ant, nothing else. And what you want to do is you want to try to make sure, you have to test to make sure it doesn't accidentally pick up the DNA of other species, right? You don't want it to amplify other ants. So I did some research and I tried them out, and I was able to find that my primers only did bind to the right type of ant, the Solenopsis invicta. And so then what I did was I took that DNA that I had amplified from their stomachs of these tiger beetles, and I was able to do something where you try to match it to a reference library and you find out what is the closest match, just to verify that in fact I did amplify the right ants. And I find this, right? You can see there was a 99% match of a population of Solenopsis invicta from Mississippi. And that's exactly what I was looking at. So that's really great. So now I've actually been able to basically prove that I was able to make genetic markers that can tell us whether or not the stomachs of these tiger beetles have fire ants in them. And then when we started looking, we found that certain populations of these tiger beetles have eaten tons and tons of fire ants. So it's not something that they eat once in a while. Some of our populations, 90% of the tiger beetles had eaten a fire ant recently. So that's really fantastic. So perhaps we can use this as a biocontrol agent. Maybe we can actually rear these tiger beetles and release more of them. And we're not introducing a new predator. They're already native to these areas. So all we're doing is augmenting their numbers potentially. So very excited about this find, and we'll see what we can do with this. Okay, and I would like to do the rest of this talk about conservation and rare species, including a species that's found in the New Jersey Pine Barrens. So this is a little table here from the International Union for Conservation of Nature, right? This is basically the red list of threatened species. This is showing how many different types of mammals, birds, reptiles, etc., are endangered or threatened, which means that they have some sort of a legal status somewhere around the world saying that these are things that are so rare and they're going extinct that they should be protected, right? And so you can see here for some of these groups, there's thousands of species of mammals and birds that are endangered. And if we look at the total number of species, we see there's about 5,500 species of mammals, and 21% of them are considered threatened. Almost a quarter of all mammals are threatened with extinction. That's really scary, right? Birds, it's about 1 in 10. Reptiles, it's about 7%. Amphibians is terrible. Almost a third of amphibians are actually endangered or threatened, right? But I really want to point out insects here. So you can see there's about 1 million species of insects, and less than 0.01% of them are considered threatened. Is that because insects are so good at avoiding being endangered? Or are they just really resilient? Or is it because we just don't tend to study insects to conserve them? Or we don't really like insects that much, so we don't tend to prioritize them? And I would argue there's a lot of reason to believe it is the latter. It's not that fewer of them are endangered. Tons and tons of insects are going extinct, but we are way more likely to care about some cuddly mammal or pretty bird than we are about some sort of a fungus beetle. That's generally the way it works. And so it's very unfortunate, but insects deserve more protection than they get. We just tend to not care. But we care about some of them. We care about them if they're pretty. Are these the only insects that are endangered? Absolutely not. But these are the ones that are going to get legal protection and the ones that are going to make these lists because they are pretty. We have the Carter blue butterfly in our upper left. Absolutely stunning butterfly, right? We've got the monarch butterfly in our lower left. Although it is not actually protected yet, in 2013, it was proposed for listing because 90% of the population had declined in less than 20 years. I don't think they're actually going to get listed, but at least it was proposed. In the upper right, we have Heinz emerald, which is a beautiful dragonfly that lives in a very specific habitat of these calcareous fens that they say, these limestone wet areas in the upper Midwest and nowhere else. And then on the bottom right, we have a tiger beetle, which is the Ohlone tiger beetle, which only lives in a small area in California. It wasn't even discovered until 1987, and most of its habitat is completely gone. It's now housing developments, agriculture, and it's really hard to find them. They're only found in a small area of remnant California coastal prairie in Santa Cruz County. They basically have an address. There's only one little area left where they occur. And this is a beautiful species. And now that it's known, it does have some legal protection as on the Endangered Species Act. But one of the reasons a lot of tiger beetles are going away, why they're not doing well, is because we don't have fires like we used to. Yeah, I know California does, but in the East we don't have fires like we used to. Fire is a natural process, and fire can be a very good thing. Obviously, excessive fires like we're seeing in California, that's another whole story, and there's different reasons why they're so bad right now. And sometimes fires are worse because there haven't been regular fires. Fire is a natural and regular process. And if you suppress fire for a long time, a lot of fuel builds up, and then fires can be hotter and much, much worse and harder to control. And it's very hard to get people to listen to that. But fire is a natural part of the New Jersey Pine Barrens. And many of our rare species in the New Jersey Pine Barrens are going to be associated with areas that recently burned and then opened up and there was bare sand. This particular species, the ghost tiger beetle, is one that I'm studying right now with a couple of colleagues, Joe Smith and Kyle Rossner. We're looking at their habitat requirements and their distribution in New Jersey. They are hard to see. I think they're beautiful when you actually get an up close look at them, but they're really hard to see because they're almost totally white and they live on white sand. Now, one of the problems they have is that their habitats go away all the time naturally. Their habitats get lost to succession, meaning over time, bare ground gets colonized with grasses and with herbaceous plants, and they start to fill in, and then shrubs and trees come in, and ultimately they get completely stabilized and all of that bare ground goes away. But naturally, fires would open up areas on a regular basis, and they would always be moving around from little population from one little area to another area. And we can call this a metapopulation, where it's all these little patches that are connected, and none of them are stable by themselves, but the whole group of them is stable because as one habitat patch goes away, the beetles are already flying out of there into new patches that are nearby, and there's always this shifting mosaic of different areas opening up, and they can kind of escape in time and space, right? So they're always going to be moving around. These little patches will get lost, but they can get around. Now, when you have housing developments, agriculture, strip malls, and a lot of these areas are being developed quickly, as those go away, it gets harder and harder for the remaining populations to reconnect to each other. And now they're isolated and they're more stuck apart. And they may go away on their own because they don't have the ability to recolonize any adjacent areas. You can't recolonize something that's now asphalt. And so this could be a real problem for this species, is their inability to find new habitats. So my colleagues and I, as I mentioned, Joe Smith and Kyle Rossner, we all in 2014, 2015 went out and we looked for tried to find as many populations as we could in New Jersey. We looked at Howard Boyd's great guide to New Jersey tiger beetles, and he had listed 18 populations of Lepida in New Jersey. We now know that there's about 11 left. I mean, it's possible we could have missed one, but we thoroughly searched all known historic sites, and we used Google Earth to find large sandy areas and searched all of those as well, even if they were never recorded to have Lepida before. And in fact, we found a few new populations doing this. But we found that about half of Howard Boyd's sites from 1978 were gone. They were gone either because of apparent fire suppression or because of some sort of developments. Those are different reasons why they might be gone. And so from these populations, not all of these were really healthy populations. A number of these appeared to be very small, very, very small, incredibly small number of individuals. And really, only the ones in red were large populations. And so we believe that each of those black dots within a circle containing a red one, those may not be actual viable populations, but they might be what we call in ecology sinks. They may be areas that have a little bit of habitat, and maybe it's not perfect, but they can survive there for a little bit. And so these big populations are always throwing out propagules. They're always producing more Lepida, and some of those Lepida will fly around and colonize these adjacent areas, but they'll never be big populations themselves. And so these big source populations are especially important to preserve because they have a lot of open habitat. We don't want those to get lost to succession. We don't want those to turn into forests, because if we do, we're going to lose a very large percentage of our remaining lepida in New Jersey. So some of these are along the coast. You can see we have 3 large populations along the coast, and we have 3 large inland populations. And they have different processes that are keeping them open. There's all kinds of beach dynamics that keep sand moving and sand open due to wind and water. And the inland ones are probably more due to fire. And so that's going to be different challenges there for conservation, something that we want to think about. And certainly fire management is a big part of the New Jersey pine barrens. So that one large population that's got a couple of little satellite sinks around it, that is a large managed area where they are found. And in fact, that's part of Fort Dix. So that is, they're doing they're doing a lot of bombing, and they're doing a lot of raking, and they're doing a lot of different things where they're keeping open sand areas. And as a result, there's a lot of lepida there. So it's a really great population. The military often does a really good job of doing conservation, honestly. And when we talked to them, they were very open to maintaining these areas for the beetles. So one of the other things that we did in this study is Joe Smith took the lead on this one. He went out to a new population that was discovered. This is the one down in Wildwood Crest, and actually surveyed for them along man-made areas where they put up fences, these drift fences that actually create dune habitat, and found that they do like man-made habitats, and they're quite happy to be in these places in similar numbers that we were finding in a nearby adjacent large population that was in a natural area. So that was very interesting. And so there's, again, implications here for it might be possible to manage to create habitat for these beetles that is also going to be a goal of beach stabilization anyway. This is something that often people want to do to maintain sand on these beaches. And so that's what I've got for you today. I'd just like to thank my many co-authors who worked on different aspects of this project with me, these different projects with me. And a lot of these fantastic photographs were taken by Jason Sepka, who's just really skilled. Ted McRae took some other photographs. Gift Beaton took some. Chris Worth and Kyle Rossner took others. And so this was a really, really great group effort here to come up with these really wonderful images. And so with that, I'd like to leave a few minutes to take some questions. So you can call in there, and I'll be happy to answer any questions that you have about tiger beetles.
Thank you very much, Dan. Dan, that was fascinating. I really enjoyed kind of real world implications of DNA and genetics, and how the science really does matter, particularly with the different populations. That was a really great to see how you were able to illustrate that.
Great, thanks.
I live down in Tuckerton, and I'm very familiar with that area down at the end of, or what's known as Seven Bridges Road, or the Great Bay Wildlife Management Area. And I could see that looked like pretty close to one of those populations there on the shore.
Yeah, absolutely. Yeah, it's a great area because it appears to harbor a very large population. And, you know, it's good to have these reservoirs of really big numbers.
Yep. Yeah, that's you know, an area that's pretty much for the most part preserved, and nothing's ever going to change down there. So that's good news to hear. All right. As Dan said, you guys are welcome to call in. The number's on the screen. It will take a couple minutes for your call to go through and for you to put the ID in, but we'll be here holding for your questions. All right, we have a caller. I will limit them. Hello, you're live on the air with your question.
Where can I get a copy of his book? Do they have it at BioQuip? Don't know what BioQuip is.
That's a great question. I don't know if it's at BioQuip. I believe it was at one point, but you can get it on Amazon. Although I think that it may be out of print now, which is unfortunate. It's 2015, and I think they sold out of most of what we had. But there are used copies available for sure. So I would try Amazon. And I would try like Abe Books, if not Amazon. Abe Books almost certainly has it.
Abe?
Abe,
Okay,
A-B-E.
ABE books.
Yeah, Abe, like
Okay.
Abraham Lincoln.
Okay, thank you.
No problem.
Okay, and we have another caller, I'll admit. Hello, you're on the air with your question.
Hi, good morning. This is Becky LeBoy from Ocean County Soil Conservation District. Thank you
Hi.
so much for this wonderful presentation. I've learned so much about tiger beetles.
Great.
Fascinating. And Dr. Dan Duran, you're such a great speaker. So you actually made it really fun to learn about the tiger beetles as well as informative. So
Oh, thank
my
you so
question
much.
is, you're welcome. My question is, regarding tiger beetles, historically at Island Beach State Park, can you talk a little bit about that? And are there any efforts in the future that you can anticipate for restoring any populations of tiger beetles at Island Beach State Park? And
Island
if
Beach
it's
State
okay
Park
with you,
is in Monmouth County, right?
Ocean County.
Ocean County. Okay. Okay. So I'm just, for some reason, I'm drawing a blank. Is this near on the other side of the island from Holgate?
It's north. Yep, north of LBI.
Yes. Okay. Oh, right. North of. Okay, I know where you're talking about. Yeah. So historically, that area had two rare tiger beetles. It had Habroselomorpha dorsalis, which used to be called Cicindelidae dorsalis. You can blame me for changing the taxonomy. So a lot of people are like, "Stop changing the names." I'm like, I only changed them once. But anyway, Habroselomorpha dorsalis is historically known from that area. It's very rare, and it is in fact federally protected. And I don't know what the quality of the habitat is there because I haven't been up that way in quite some time. But if there's large coastal beaches with back dunes and they're not regularly raked and it's not driven on, there's always a possibility of restoring dorsalis to an area like that. So that would be great. And it's actually worth just looking to see if they're there naturally. I don't believe anybody's searched that area in years. We know they're in Holgate and they're in Little Beach. But north of Holgate, I don't know of a single population that is extant, but they could be there. So it could be worth looking, in July, August this year for sure.
Great. Thank you very much for the information. Keep up the great work.
Great. Thank you.
All right, thank you very much for your question. Yeah, that's a great area there, Island Beach State Park. It's basically everything south of Seaside, and it runs the whole way down to Barnegat Inlet. So there's a pretty large, undeveloped area with lots of dunes and beach. There is probably some driving that occurs there because there's some recreational fishing down in that area, but it probably would be a great place to investigate a little bit further.
And if
for
nothing
those
else, the
populations.
dunes. Oh, I'm sorry. I didn't mean to interrupt. Sorry.
No,
Go
that's
ahead.
okay. No worries.
If nothing else, the dunes may have lepida. I mean, they were known from, large, some areas up towards Ocean and Monmouth County up there. And that could be worth looking there for sure. I have not searched that area very well, but one of my colleagues has, and he didn't find any, which is why we didn't go back looking into that area. But it's quite possible that he may have missed them and they could still be there.
Yeah, I mean, dune-wise, it's probably one of the most prolific dune, because it is undeveloped. There's a road, but there are lots of natural duned areas. So there probably would be a good opportunity there.
Yeah,
To
great.
check them out. Little Beach, I love Little Beach. My grandfather was born on Little Beach. My great-grandfather was worked for the Life Saving Service, and that's where he was stationed, out there
Oh, wow.
on Little Beach.
Oh, very cool.
Yep. I spent a lot of time in the inlet, in the bay down there, where Barnegat Bay meets Great Bay. And, there's no houses or no people out there, but there once was a little community. But I mean, talk about a perfect spot for wildlife and nature. It's basically been completely reclaimed by nature, being that there's no structures left. And really, there's nobody out there anymore.
Well, and
but
that's,
it is a neat place.
yeah. And that's why that's the last place for a wild population of Habroselomorpha dorsalis. And they're all over Little Beach. Like there's a good, healthy population there, and nobody drives on the beach. That's it. You can't
Yep.
drive on it because you can't get to it by car. So.
Yep. Yeah, and that talk about an isolated population. At least there's room there for plenty of them to survive, but there's probably not a great chance they're going to be able to go anyplace else because they're so isolated there, basically on an island, in the inlet.
Yeah, and they get to Holgate because of the the south end of Holgate being protected. So I've seen them in Holgate too. It's all spillover from Little Beach, and they probably once in a while show up in North Brigantine, but that
Okay.
area is too heavily traveled, so I don't think they could ever persist there.
Would they be transported by the wind and by the water sometimes, or just by going for a long flight?
Long flight, they can move up to 30 miles on their own power in
Oh,
a
wow.
year. Yeah, they're quite capable, and that's why if Island Beach State Park is really great habitat, I mean, some of these beetles may find it, and we should probably go search there again. Maybe this year I'll get to go out there, and
Yeah.
anybody that wants to do that, contact me, and maybe we could spend a little bit of time looking.
Yeah, I mean, I think LBI itself is basically 18 miles. So the one end of Holgate to Barnegat Light would be 18 miles. Just across the inlet over there would be the end of Island Beach State Park. So definitely within that 30-mile range for sure.
Yeah.
All right, we have another person calling in. Hello, you're live on the air with your question.
Yes, I have a question for you. The current administration has made some changes to the Endangered Species Act, and I'm just trying to find out if there's any concern that those changes that are now, I think they're now law in the Endangered Species Act, can have an effect on some of the tiger beetle habitat here in New Jersey, the way they're redefining things.
Yeah, that's a great question. The short answer is yes. There have been changes, and it is harder to protect endangered species now. It's easier to get around the protections that were historically in place. I hope that some of this will be reverted with a regime change here. So that's a good question, and I'm not actually 100% sure of all the details of that. I do know though that it has made it easier for people to develop areas that were previously protected. So the good thing is the state of New Jersey, I think, does a really good job of doing conservation, and you have areas that are protected that do tend to be kept. Some of these places are, like Joel was saying, they're in great shape, and they're being maintained. And you have multiple partners here: the Fish and Wildlife Service, sometimes private Nature Conservancy, and I hope that we're going to see that it won't affect New Jersey as much as it might elsewhere.
The other question I have for you is the state of New Jersey has yet to replace a couple members on the Pinelands Commission. For some reason, the Senate refuses to have the hearings to put Governor Murphy's representatives on the Pinelands Commission. Is that lack of those folks being on the commission affecting any type of preservation work in New Jersey, particularly in the Pinelands right now, that you're aware of?
Well, I that's it's a little bit too political for me to comment on. I understand your question. I totally do. But
my point is, are there things that are currently not working or not going on just because they don't have a quorum to vote? That's that's just holding everything up, I guess.
I can step in a little bit there, Dan.
Okay.
In those cases, the members who were on the term prior still sit. I think there may be one case, that one vacant seat right now, but on those other instances, the person who was in the position before would still be in the position until someone else is appointed. So it hasn't really affected the ability of a quorum. I would say that it's not limiting the commission to act, but it is that would be the answer there.
All right. Well, thank you very much.
Thank you for your question.
Thanks, Joel. You know a lot more about this than I do.
Well, yeah, no problem. I mean, that's one of those things. That's legislation, laws, governments, all that can change. So that's, one really important reason why we do educate, and we try to make people aware of the situation, because absolutely things can change in the future. If the political winds shift, some of the protections and things we have in place could go away. So, another reason to stay kind of alerted and to stay in tune to what's going on, and raise awareness, and just allow people to, be familiar with all that's taking place. Okay, Dan. Well, I want to thank you. Again, I was fascinated with the program. I really enjoyed kind of the deep dive into the scientifics and the DNA and the genetics. I really thought that was great to see those real world applications. And I appreciate your time, and thank you very much.
Great. No problem. This was fun.
All right. Well, with that, I'm going to shut down the live stream, and I'll talk to you on the other side.
Okay, great.